Okay, so today I'm going to talk to you about transforming cancer cells into microbe mimetics, which basically means making cancer cells look like bacteria so that they're immunogenic. So our target is ovarian cancer, and unfortunately, most women are diagnosed at late stages, at stage 3 or 4. When women have stage 4 cancer, there's only a 17% chance of a 5-year survival rate. The medical costs associated with ovarian cancer are about $100,000 or more per year. Currently, the standard of care for ovarian cancer is surgery followed by chemotherapy. And despite an improved clinical response initially, most people relapse and succumb to and basically die from the disease. So currently, there is an unmet medical need for therapies that elicit durable responses for patients with metastatic cancer. In the tumor microenvironment, there's immune suppression. So we really need immune therapies that can create an anti-cancer immune response that can activate that suppressed tumor microenvironment. So what we're doing is we're creating a personalized therapy, which means that we're taking the cancer cells from a patient and we're engineering them so that they look like microbes. This allows the cancer cells to engage immune cells and activate receptors on the immune cell surface. So ovarian cancer begins in the ovary, but quickly metastasizes, or in late stages, metastasizes to the peritoneal cavity. Within the peritoneal cavity are immune cells as well as abundant cancer cells. In late stages, women develop fluid in the peritoneal cavity known as ascites. What we're proposing is to take that fluid, which is rich in cancer cells, a bunch of, basically a large group of them exist as cancer spheroids, which in the image here are clusters of cancer cells. And one of the benefits of this is that they rapidly basically settle out by gravity filtration. And we can actually use a filter to even basically enhance purification of these cancer cells even more. Our process involves cryocell silicification and basically decoration of the cell surface with these microbial associated molecules, which in this case are toll-like receptor ligands, specifically PEI, CPG, and NPL, and all these are associated with bacteria. Our process is rapid. We can do it within 24 hours. And as I said, it basically leads to immune activation and cancer cell killing. So as I said, our target is using the societies to create the vaccine. So it is for women that have stage 3 or 4 cancer. Our process, as I said, is less than 24 hours to basically take the cancer cells and transform them into a vaccine. The surface is basically stabilized with silica so that these cancer cells become non-viable, but they become stable and they maintain the cell integrity. We decorate the surface with these pathogen-associated molecules. And what's interesting is because we've also stabilized these cells with silica, we can actually dehydrate this for shelf storage so that it does not have to be stored at -80 or in liquid nitrogen. We found that localized delivery of our vaccine works best, so actually delivery into the tumor microenvironment to actually activate those immune cells. And 2 doses works great, a prime and a boost dose. And showing here, if we actually target an animal that has late stage, meaning it's got cancer spread throughout the peritoneal cavity, we can actually have synergistic effects between standard of care chemotherapy and our vaccine. So one thing that we're doing by putting these microbial molecules on cancer cells is actually really attracting those immune cells. So we're looking at here by flow cytometry is uptake of these immune cells, of this vaccine by immune cells. So when we put these molecules on the surface, we get abundant uptake. In contrast, when we take live cancer cells or fixed cancer cells, these immune cells pretty much ignore the cancer cells with very low uptake. And this is a picture of a dendritic cell that's basically a 3D image that's been cut open. And this is the nuclei of the dendritic cell. And this is the nuclei of our vaccine cell. So this is a vaccine cell that exists and it's been eaten by this immune cell. These are called dendritic cells. And what they do is they take these tumor antigens and they process them and they present them to T cells. So shown here in green is our dendritic cell. And then these are T cells being activated by that dendritic cell that's had the vaccine. So these T cells then go out and they find the cancer cells and they induce apoptosis, causing the cancer cells to die. And the benefit of this is they can go throughout the body and find metastatic as well as localized disease. Our vaccine, having these microbial molecules on the surface, activates immune cells. So we see an increase in activated T cells, both CD4 and CD8. We see a decrease in suppressive T cells, which are Tregs, as well as T cells that express these immune checkpoint inhibitory molecules, such as PD-1, which you've heard a lot about, I'm sure, if you're following the immunotherapy. what we also see too is an increase in our effective memory T cells. So this is some of our preclinical studies where we're using a mouse model that has metastatic ovarian cancer. And we're using bioluminescent cells to follow the cancer cells. So what you see here are the cancer cells. And as they become red, they're growing and expanding in the peritoneal cavity. So when we don't treat our animals, within 25 days, basically, these animals succumb to the disease. However, when we vaccinate these animals, we're actually able to eliminate 100% of the vaccine when we treat them at early stages. And so this is creating a vaccine from both a cell line as well as from ascites that's taken from an animal with cancer. And what's really cool is that if we don't do anything else to these animals, but we give them cancer again at a later stage, they rapidly get rid of that cancer. So the cancer cannot engraft in these animals, which is basically evidence that they have these memory T cells that knock out the cancer cells. As I said before, one of the things that solidification does is it stabilizes the cells. So what we can do is we can actually dehydrate these cells. When we take an irradiated cancer cell and dehydrate it compared to our solidified cell and dehydrate it, and then we try to resuspend it in PBS at a later stage, in this case it was 2 weeks, we can resuspend our solidified cells, whereas we can't resuspend the irradiated cells at all. They're basically not stable enough. And so this is just showing you that if we actually take these dehydrated cells and rehydrate them and make a vaccine, again, we're able to get rid of the cancer in these animals. And it was, the results were non-significantly different from the fresh vaccine. So they both worked equally well. Our competition in the market for ovarian cancer vaccines are ones basically use attenuated mycoplasma, which is BCG, which has problems in terms of heterogeneity, as well as the toxic molecule dinitrobenzene. This another company at UPenn had basically a vaccine that used tumor cell lysate. And one of the benefits of a whole cell is that it gets uptaken by these immune cells. When you just have the lysate, it gets degraded in the serum by proteases. And this actually was terminated, this study. Another clinical trial, or two other ones, both at Penn Israel and UPenn, were basically taking either blood or bone marrow cells from the patient, differentiating these cells to create basically dendritic immune cells. So these are lengthy processes that use basically in vitro cell culture, cytokine, DMSO, cryopreservation, as well as this one actually uses LPS, which is an endotoxin for stimulating the cells. This technology is protected by actually 3 patents that are actually on the cancer cell vaccine. Our solidification process also results in stable genomic material and proteomic material within the cells. So we have a patent on that as well, a patent application. The leads on this are myself. I am a research assistant professor in internal medicine, and I have an adjunct faculty position in chemical and biological engineering at the University of New Mexico. And I also have an affiliate associate member position at Houston Methodist in Texas. My main collaborator is Sarah Adams. She's an associate professor in obstetrics and gynecology, and she is a clinical oncologist who sees women who have ovarian cancer. Sees and treats. So as I said, our process is a rapid process that we can actually do within 24 hours. We create these vaccines, and actually for clinical trials, you can actually create these vaccines in collaboration with existing cell processing centers. The main cost of our vaccine is those toll-like receptor ligands that are on the cell surface. So our typical vaccine is about 3 million cells, and the cost of the CPG and the NPL is about $5.6. And if you looked at 2 to 6 doses per patient, that would be about $33 for the cost of creating the vaccine without the overhead costs, which comes out to about $705,000. $705,000. Wait, that sounds $705,000. Yeah, and if you actually considered overhead and stuff per patient, we're talking about a market that's about $209 million. So takeaways from this are we are creating a personalized therapy. That means it's tailored to the patient using their own cancer cells to attack that cancer. The adjuvant that's on the cell surface, meaning those toll-like receptor ligands, can actually be tailored to the patient and their microenvironment. We've shown high preclinical efficacy and that our therapy actually synergizes with other cancer therapies. Our process is rapid and biocompatible. We can dehydrate for easy storage and transport, and vaccine production can be performed at existing cell processing centers. And down here is my email and phone number if you need to contact me. Thank you.
Great, thank you very much, Rita. I'll open it up to our panel for questions. So please, feel free to ask any questions to Rita.
I'll go first if it's okay. So, hi Rita, nice to meet you. I'm Kirsten. Really fascinating technology. That was, and obviously very well thought through. And the animal model data looks fantastic. You mentioned the cell processing centers. So do you imagine then having a startup company then that would be producing the materials, et cetera, providing that to the cell processing centers, and they'd then be customizing in this per patient, and the patients would be directed to the cell processing centers to have this happen?
Right. So I've actually gone to Colorado and I visited some of these cell processing centers, and I've actually talked to them. And what they would like is for our team to go out there and train them on what to do. And they would actually perform the studies there themselves. And they actually get this material for other types of studies. So this could be integrated into work that's already ongoing there.
Okay.
Hey, Rita, it's Mike. Nice presentation. Maybe just building on Kristen's question. I wasn't really sure of the business model then. Where are you going with this from a business perspective? How would investors view the company and the opportunity? Walk through a little bit more. Just your early thinking there.
So actually, currently, I am just a scientist right now. I actually do not have a company at this time. So I'm actually looking for business people who are interested in this product that I could work with in terms of being the scientific lead on this project.
Hi, Rita. Is this technology, can it be applied to other types of cancer, or is it specifically to ovarian cancer?
So actually what we're doing right now in the lab is we're actually testing this in both solid tumors for melanoma as well as breast cancer. And we've shown efficacy in breast cancer, and melanoma is a little more challenging. And so what we're doing is we're actually doing studies using also checkpoint inhibitory molecules as well to enhance the technology. And we've also got a study going on where we're actually using humanized mice. So we actually take mice, we give them a human immune system, then we can inject human cancer cells and use a human vaccine to validate that this would actually work with human cells. Okay. And we just started that about 2 weeks ago, so that one's ongoing.
Great.
Yeah, that's helpful to hear, because I think both, following up on Mike and Matthew's comments is, as an investor, we're looking for, what is the model? What are we investing in? What are the, potential assets, opportunities where these assets can play into? So actually knowing about that, the other applications is incredibly helpful towards perhaps then, being able to approach an investor and say, here's what we want to do. Can you help us form a team around building the business model for this? So that was really helpful to hear.
Exactly. And our clinical people would love to have clinical trials here at UNM as well.
Good plug.
Rita, do you think that you would do a joint venture with a large pharmaceutical company? Or I guess what's your vision for, the next step on the company?
That would actually be fantastic, because that way they have the funding, we can actually do this and jump right into the clinical trials, which is what we're very ready for.
Okay.
And have you approached partners, Rita? I mean, you mentioned looking for, I'll call it a business driver. That may not be the right term here if you're looking for corporate support, but kind of who's interested from a corporate perspective, do you think, given the competition?
I have not talked with big pharma directly. I've talked with some smaller companies. There has been actually a lot of interest in this, but I really wanted to get the data out first. And so we recently published this data in Nature Biomedical Engineering. So we are ready to go now.
Just any sense of how much capital you would need to deploy over the next couple of years to move this forward, just high level?
I was estimating for safety studies, maybe about 5 million to get in and actually show that we can do the right doses and things like that. Yeah. Mm-hmm.
Right. Do we have one last question and then we'll wrap it up? Right. I think we're done. Thank you so much.
Thanks, Rita.
Thank you.